Hello, I'm Saida Kamar. Today we are covering Chapter 4, Section 6. I will be emphasizing on the differences between DNA and RNA. DNA and RNA have several differences. The first is their location. Looking at any individual cell, the nucleus consists of chromosomes, which are made up of DNA. RNA is found in the cytoplasm of the cell. Another difference between DNA and RNA is their structure. As you can see in the image, DNA is double-stranded. It is also called a double helix. Because of the helical shape its strands form. RNA, on the other hand, is single-stranded. DNA and RNA also differ in the 5-carbon sugars they use in their genetic makeup. DNA uses deoxyribose, while RNA uses ribose. This is also the basis for their name. Deoxyribonucleic acid. Ribonucleic acid. The nitrogenous bases used in DNA and RNA are slightly different as well. DNA uses cytosine, guanine, adenine, and thymine, while RNA uses cytosine, guanine, adenine, and uracil. As you can see in the image, the first three are the same, while the last two are different, the thymine and the uracil. The functions of DNA and RNA are also different, but very important because they work hand in hand. DNA contains the genetic code needed for protein synthesis, while RNA transcribes and translates to form proteins. The whole process begins in the nucleus, with DNA. A strand of DNA replicates, forming the double helix structure, as you can see here. At this point, the function of RNA comes into play. So there are 3 forms of RNA: messenger RNA, ribosomal RNA, and transfer RNA. After replication of DNA, one of the strands is used as a template for RNA. This process is called transcription. The messenger RNA carries information, or the transcribed template, from DNA in the nucleus to ribosomes in the cytoplasm. The ribosomal RNA forms the structure of the ribosomes. After the transcription process, the transfer RNA carries amino acids to the ribosome to translate the transcribed strand to help build an amino acid chain. This process is called translation. This is where codons are translated into proteins. Remember, codons are sets of 3 nitrogenous bases that translate to a protein. So for example, these 3 nitrogenous bases here would convert into this protein. This concludes the differences between DNA and RNA. Thank you for watching!